Good evening, everyone. I welcome you all to the Week 5 live problem solving session on NPTEL Bioinformatics Algorithms and Applications course. So, in this week, we're going to deal with the Week 5 concepts, so which includes uh, discussion about the redundant and non-redundant sequences and how to get a non-redundant sequences from redundant sequences, what are the tools or techniques that are available, and followed by that we will be discussing about the protein secondary structure, followed by the tools, computational tools or methods that are available for secondary structure prediction. So in bioinformatics, redundant and non-redundant datasets are terms used to describe the collection of biological sequences. Biological sequences are nothing but the collection of proteins or nucleic acids. So the name redundant and non-redundant is given based on their similarity to each other, sequence similarity to each other. Redundant datasets contain sequences that are highly similar or identical to each other. These redundant datasets often include multiple copies of same protein or sequences that share high degree of identity among themselves. For example, we know UniProt, which is a comprehensive resource for protein sequences and functional information, where when we observe UniProt, UniProt has two parts. One offers redundant and other offers the non-redundant dataset. When we observe the UniRef option of UniProt, which provides the non-redundant sets by clustering the sequences at different identity thresholds. So today we will be seeing how a redundant dataset will be converted to a non-redundant dataset using the clustering techniques. Then comes the concept of non-redundant datasets, which contain sequences that are sufficiently diverse and dissimilar from each other. These datasets aim to represent a broad range of biological diversity while minimizing the redundancy to facilitate the computational analysis such as sequence searches, alignments, and structural predictions. So, why we need non-redundant datasets? Non-redundant datasets often lead to improved performance in data analysis and machine learning tasks, and these redundant data can introduce bias and skew results, whereas a non-redundant dataset provides a more balanced representation of the underlying data distribution, leading to more accurate prediction models. Non-redundant datasets require less storage space compared to the redundant datasets, which can be especially beneficial when working with the large datasets. Here, by removing the duplicates or highly similar entries, the dataset size is reduced without sacrificing the important information. Without redundancy in a dataset, it is easier to identify and interpret meaningful patterns and trends, leading to more informed decision making. Non-redundant datasets can lead to faster model training times as there is less data to process and fewer redundant patterns to learn. This can be particularly advantageous when working with the large-scale machine learning models or when computational resources are limited. Overall, we can tell that non-redundant datasets plays a crucial role in ensuring the quality, efficiency, and effectiveness of the data analysis and machine learning tasks across the various domains. So to understand how a redundant and non-redundant dataset, so here I've given a collection of some geometric objects. So based on this, can you guys answer what is a redundant dataset and what is a non-redundant dataset? So given here two sets of data which has a collection of geometrical objects, what is a redundant dataset and what is a non-redundant dataset?
B is non-redundant.
Yeah. So moving forward, how a redundant dataset is converted into a non-redundant dataset. First, given two datasets, we try to focus on dataset A. So from dataset A, we'll try to group the similar elements together. So here I am grouping similar geometrical elements together. So here the first Three circles represents one cluster or one group. Next, three rectangles represent another cluster or group, followed by cylinders, triangles, and rhombus. So here the grouping is performed based on their similarity among themselves. Here all are circles, all are rectangles. Here all are cylindrical shapes. So here I have used different color codes to tell that these shapes may be similar, but they lack some identity among themselves. That is, they are similar, not identical. Even in this case, next, once these clusters are built, now we have 5 clusters. So from each cluster, a representative sequence has been selected. So here I am selecting one from each cluster to form a representative data set. So this representative data set is termed as a non-redundant data set, whereas A is a redundant data set. So this is a simple example on how a clustering is performed on a redundant data set based on similarity searching. Then on each cluster, a representative of a cluster is selected to form a non-redundant data set. Further, we'll see how biological sequences are used in this process to build the non-redundant data set. So first, and the popular method is CD-HIT, which refers to a widely used bioinformatics tool for clustering and comparing large sets of proteins or nucleotide sequences. Here, CD-HIT stands for Cluster Database Set High Identity with Tolerance. It was first introduced in 2004 and has since undergone several updates and improvements. CD-HIT works by identifying a group of sequences that share high degree of sequence similarity and clustering them together So this is what we did in our previous example. So we found what share high similarity, then we clustered them together. Then it emerges out single representative sequence from each cluster. So this CD-HIT can be useful in many applications, such as identifying homologous genes across the species, reducing redundancy in large data sets, and identifying potential function domains within the protein sequences. So how CD-HIT works? CD-HIT takes a set of biological sequences as input, then CD-HIT employs a fast and memory efficient algorithm to calculate pairwise sequence similarities. Even we have studied in previous classes how pairwise sequence similarities has been calculated. So here CD-HIT has many ways to find this pairwise sequence similarities. One such method is word-based approach or k-mer counting approach, which is a part of BLAST algorithm that we have discussed. So why I am discussing, because this is one of the method that helps to rapidly identify the potential sequence matching before performing a computational intensive alignment calculations. So the similarity between two sequences is usually measured by sequence identity, like which is the percentage of identical positions between the two sequences, that is number of in the alignment number of identical residues that are present divided by the total length of the sequence. Then it comes to clustering. So based on the calculated sequence similarities or identities, CD-HIT performs a clustering process to group the similar sequences into a cluster. Here, clustering starts by selecting a representative sequence. In general, this representative sequence will be the longest sequence by default, as a seed for the first cluster. Then it iteratively adds sequences to this cluster if they meet a predefined similarity level. If sequences that do not meet similarity threshold with any existing sequence or existing cluster, then this becomes the seed for new clusters. The clustering process also I will be discussing in detail next. Then here CD-HIT outputs a non-redundant set of sequences where each sequence represents a cluster, and this reduces the overall size of the sequence data set while preserving its diversity. CD-HIT provides several output files including clustered sequences, a file specifying the cluster representatives, and statistics on clustering process. Users can further analyze the resulting clusters for various bioinformatics applications such as sequence annotation, phylogeny analysis, and functional characterization. When coming to the clustering, particularly the clustering process in CD-HIT, it uses a grading incremental clustering algorithm method, wherein sequences are first sorted in order of decreasing their length. That is, longer sequences first, the shorter sequences at the last. The longest one becomes the representative of the first cluster. Then each remaining sequence is compared to the representatives of the existing clusters. If the similarity with any representative is above given threshold, it is grouped into that cluster. For example, we made a cluster with the longest sequence, then we take the second longest sequence and compare it with the longest sequence. If it is having similarity more than the defined threshold, it is added into that cluster. If it is not having the similarity, or it is having the similarity below the threshold level, it is seeded to a new cluster. So the first step in clustering is divided into three parts. Clustering is divided into three parts. First is clustering initialization. So as we discussed, it selects a seed sequence as an initial representative for the first cluster. So this seed sequence is often the longest or the most abundant sequence in the data set. Then CD-HIT iterates through the remaining sequences in the data set and adds them to the existing clusters, or creates new clusters based on their similarity to their existing representatives in the clusters. As the clustering progresses, CD-HIT updates the representative sequences for each cluster. The representative sequence is typically chosen based on criteria such as sequence length, abundance, and other user-defined preferences. So when we compare general clustering methods with CD-HIT, this table provides a differentiation between these two. General clustering methods operates on numerical or categorical data, treating each dimension equally without domain-specific knowledge. Whereas CD-HIT operates on biological sequence data, considering the sequence identity or similarity, and there are some parameters which are optimized for bioinformatics applications or bioinformatics driven results. So when we go for general clustering methods, they use a variety of algorithms like hierarchical clustering, k-means clustering, etc., which often rely on distance metrics. So when we come to CD-HIT, which employs a fast and memory efficient algorithm, combining the word-based and alignment-based method tailored for sequence analysis. In general clustering methods, each cluster is represented by a centroid or a characteristic feature summarizing the data points within the cluster. Whereas in CD-HIT, each cluster is represented by a single sequence, aiming to capture diversity while minimizing redundancy. So how are we able to capture diversity when we are removing the sequences from the initial data set? So the clusters are made such a way that the sequences within the cluster are very similar to each other, and the sequences among the clusters are very different. So why we are removing the redundancy but still maintaining the diversity. CD-HIT program has several different commands, each with its own specific purpose. We have normal CD-HIT, size, position specific, iterative CD-HIT, and many more. So here is an example of various parameters and options that can be used with the CD-HIT command. So first, the flag I denotes for input file. So here we give the input file containing the sequences to be clustered. So this input file can be in FASTA or FASTA-Q formats. Next we have O flag, which denotes for output file. The output file is where clustered sequences will be saved. Next we have C flag. So how to identify a flag? In any coding or software programs, two hyphens followed by single letter or single hyphen followed by alphabet denotes a flag term. For example, here CD hit hyphen I is a flag. So when we come to C flag, it denotes for cutoff. So what sequence identity cutoff or what sequence identity threshold for clustering to be used. So when you give a particular cutoff value, sequences with a pairwise sequence identity greater than or equal to this cutoff value will be grouped together in the same cluster. So this clustering value can be changed based on the work or based on the objectives. Next we have N flag, which stands for word length. So here the length of a word is used to compare sequences. Increasing the word length can increase the sensitivity but also increase the computational time. Next we have some computational related terms such as memory, that is maximum memory or maximum processing speed that could be allotted to this. So this D flag can be adjusted based on the available resources on the computer. If D flag is not mentioned, it will try to allocate itself the maximum allocated processors that it could CD hit command could take. Next we have clustering methods. Recently this M flag has been introduced, which is to select the clustering method. So there are three options. The default option is fast but not as sensitivity as other methods. Next we have a slower method which is denoted by AC, but the sensitivity is high. That the sensitivity is more when we use AC as a clustering method. So this method helps us to cluster sequences with low identity as well. Next we have a G flag, which is for greedy clustering method, that can be faster than the default method, but it turned to be not that accurate. Similarly, the T flag, which tells about the number of threads. This number of threads is a concept of parallel processing. So this can speed up the clustering process on multi-core computers or servers or high-end computers. Here I am showing an example usage of CD hit command. This command will cluster the sequences in the input.fasta file using a 0.8 sequence identity cutoff and a word length of 5 and a maximum memory usage of 1000 MB. So it will use default clustering method and run on 4 threads. Here the resulting clustered sequences will be saved in the output.fasta file. So here I have given an example of 5 sequences with that similarity matrix available and cutoff is 8. So here initially for simplicity I am considering all these sequences are of same residue size. So first, among this similarity matrix, we try to identify the set of sequences that are highly similar to each other. So based on the similarity matrix, 3 types of clusters has been built. So how these clusters has been built? First, when we observe A and B show a similarity of 9, which is more than our cutoff. So A and B has been grouped. Next, we try to check is there any other sequence that has more similarity than cutoff. When we see C, D, E are less than the cutoff values, similarly C, D, E for B has values of 7, 5, 2 only. Then we take up C. Since C doesn't share any similarity beyond the threshold cutoff, C has been made into a separate cluster. Finally, since D and E share the similarity of 8, they have been clustered. So finally, this similarity matrix between sequences A, B, C, D and E given us 3 clusters. So this clusters is an output file which we can see in output.fasta, which will contain the clustered sequences. So the header of each representative sequence will contain information about the cluster, including the number of sequences in the cluster, percentage of identity between the representative sequence and other sequence in the cluster. So similarly, I have given some other sequences, adding one new sequence F. So try to perform clustering on this. You can mention in the chat box a number of clusters and what are the sequences under cluster 1 and cluster 2, like that. So given 6 sequences here and similarity matrix among themselves, try to perform clustering. Remember the cutoff here is 8. So the simpler tech to perform clustering is to try to identify between what pairs of sequences are having values more than equal to or more than the cutoff. For example, I am giving one clue here. When we observe uh, A and B has a value of 8. So collect the information about the pairs of residues that share sequence similarity more than the given cutoff. Okay, I'll move. So we'll try to build a cluster followed by this. So here I have already grouped pairs of sequences that share more than sequence identity threshold. we'll solve it here. So I identified the pairs which share similarity more than the threshold of 8. So when we try to group them, so here we have A, B, C, next we have D, E, F, and when we observe intercluster similarity here, when we observe the similarity A with any one of DF, it is less than 8. B with any DF is again less than 8. C with similarity with any DF is again less than 8. So now here we have two clusters. So cluster 1 could be ABC and cluster 2 could be DF. Then so from a redundant sequences, so two clusters having ABC and DF has been formed based on similarity. Then from each cluster either any representative has been selected, finally leading to a, okay, here I am selecting the first letters of each cluster. So a redundant data set has been converted to a non-redundant data set here. Yeah, I can see one question. All are not having sequence identity of 9. For example, when we observe sequence identity of A and E is 1. So this is how the sequences has been clustered. Moving forward, we have one more software which is Spices. This is actually used for curation and filtering of protein sequences in bioinformatics, and it is primarily used for creation of high quality reference data sets for use in sequence similarity searches, protein structure prediction, and other applications in field of computational biology. So this tool works by taking a set of protein sequences and filtering them based on variety of criteria such as sequence identity, length, and presence of non-standard amino acids or gaps. The resulting dataset is then clustered using a hierarchical clustering algorithm. Previously, CD-HIT uses a greedy search algorithm. Here, it uses a hierarchical clustering algorithm which groups similar sequence together based on pairwise sequence identity. The cluster dataset is then used to generate a non-redundant reference dataset which contains a representative set of sequences for each cluster. This reference dataset can be used for a variety of applications such as sequence database searching, homology modeling, and protein function prediction. One of the key features for Pisces is its ability to filter sequences based on their structure as well as their sequence similarity. This helps to ensure that resulting reference dataset is of high quality and suitable for use in wide range of computational biology applications. Okay, previously I forgot to mention CD-HIT is available in this link. So one can click on this link. Previously there is a graphical user interface version of CD-HIT available, but for now it has been discontinued. We have only a GitHub page. So when you click on this GitHub page, all the installation instructions has been given in readme file. So in this GitHub page we also have size CD-HIT. So size CD-HIT stands for position specific iterative CD-HIT. So different Perl based scripts have been provided, and how to access this scripts is given in the CD hit file, and user cases has been also given in omics datasets. These are the example files that has been available here. Similarly, PySys is available at the link given here, and this is a graphical user interface is still accessible. So one can click on this, access the server to create your own list option. So here one can cull the sequences from whole PDB, or you can cull from your own list and you can submit the sequences or PDB IDs different. So the one problem with the PySys server is it is the only source of server that is available which takes structure as input. So because of overload, it crashes very often. So a CD hit installable version is often preferred. Yeah. So PySys, as we are discussing, this is a powerful tool for curation and filtering protein sequences and has become important component for many bioinformatics pipelines. Sorry. So when we compare both CD-HIT and PySys, both are useful tools for managing and analyzing large data sets, but they have different strengths and weaknesses. CD-HIT is particularly suited for clustering large data sets quickly and efficiently, while PySys is useful for reducing redundancy in protein databases and improving the quality of resulting data. So CD hit is a time saving algorithm, whereas PySys is a quality building algorithm. So the choice between these two tools will depend on the specific needs of the researcher and characteristics of the data that is being analyzed. So with this, I have one question. So what type of clustering algorithm does CD hit employ? So I got two different answers. One is about k-means clustering, and other is about uh, greedy incremental. So CD hit actually uses a greedy incremental clustering. This is as a default method. So wherein in this method it selects a seed sequence as an initial representative for the first cluster. So this seed sequence is often the longest sequence or the most abundant sequence in the dataset. Then CD hit iterates to the remaining sequence in the dataset and adds them to the existing clusters or creates new clusters based on their similarity to their existing representatives. So sequences are added to the clusters if their similarity with the cluster representative exceeds a specific threshold. Then we have about protein secondary structures. So proteins are complex molecules composed of long chain of amino acids. The way these amino acids are arranged in space determines the protein's overall shape, and specific shapes of different proteins are referred to as secondary structures. That is, local arrangement of these amino acids is often termed to as secondary structures. There are three main types of protein structures, that is alpha helices, beta sheets, and loops. So alpha helices are tightly coiled structures that resemble a spring. They are formed by single polypeptide chain that coils around itself, forming a helix kind of structure. Whereas beta sheets are flat and sheet-like structure composed of multiple strands of polypeptide chains that are arranged side by side. So these strands are held together by hydrogen bonds between the backbone atoms of the adjacent strands. So this is a pleated ribbon-like structure. Then we have loops, which are also termed as turns or coils. So we have a concept of dihedral angles. So that dihedral angles in protein refers to the angles between the adjacent chemical bonds in the peptide backbone. These angles determine the conformation of the protein backbone and play a crucial role in protein folding and function. Here is an image of protein backbone with dihedral angles labeled. The phi angle is between the nitrogen and the C alpha atom in the protein backbone, and the phi angle is the angle between C alpha carbon and the carbonyl atoms. The values of these angles are often plotted on a Ramachandran plot, which shows the distribution of phi psi angles in a protein. So here Ramachandran plot is a tool used in structural biology to visualize the dihedral angles, that is phi and psi angles, and these angles describe the rotation of peptide bonds between the amino acid residues. These plots helps in assessing the allowed and disallowed regions of the backbone conformations based on steric clashes and hydrogen bonding. So Ramachandran plot is divided into four main quarters or regions. The first quarter will start from the phi and psi positive values, that is right side top box. Then it moves to left, down, and again towards right. The quadrants are named like that. So the quadrant 1, this region corresponds to the combination of positive values of phi and psi angles. In this quadrant, the backbone conformation is characterized by the presence of hydrogen bonds between carbonyl oxygen of residue and the amide hydrogen of the residue, forming an alpha helix, that is I and I plus 4. Then we have quadrant 2. This region corresponds to the combination of negative phi values and positive. So in this quadrant, the backbone conformation is characteristic for, this is for negative phi values and positive psi values. So in this quadrant, the backbone conformation is characteristic of beta sheet secondary structure with hydrogen bonding between neighbors. Finally, we have quadrant 3, which is for right-handed alpha helix. This region is also known as generously allowed regions because very less observed. Both negative phi and psi angles are very less observed. So wherein this quadrant 3 corresponds to the combination of negative values of both phi and psi angles. These negative combinations are less common and they are still observed in protein structures. And finally we have quadrant 4, which is combination of positive phi and negative psi angles. The combination of these quadrants are energetically unfavorable due to steric clashes between the atoms of the protein backbone. This is how a Ramachandran plot helps to assess the quality of protein structures. So coming to the tools that can help to predict the secondary structure of proteins, the first and foremost used is DSSP, which stands for Dictionary of Secondary Structure of Proteins. Is a widely used program in bioinformatics for analyzing the secondary structure of proteins. It was first introduced by Scapes and Sander in 1983 and has become an essential tool for researchers studying protein structure and function. So DSSP works by analyzing the coordinates of each amino acid in the protein, which can be like labeled with the respective secondary structure codes. Here, the program assigns a secondary structure states to each residue based on its position and orientation of the backbone atom. The secondary structure states are classified into 8 categories: alpha helix, 3-turn helix, 5-turn helix, pi helix, extended strand, beta bridge, bend, and random coil. This program also assigns a hydrogen bond state to each residue, which can be used to analyze the stability of the protein structure. DSSP generates an output file that contains a summary of the secondary structure and hydrogen bonding patterns. So there are several methods available for predicting the protein structures, which refers to the local protein secondary structures, which refers to the local conformational arrangement of protein backbone. These methods are broadly categorized into experimental and computational methods. Experimental methods are like X-ray crystallography, NMR, many such structure prediction methods, but computational methods are of main focus because they reduce time and cost. So computational methods include sequence-based prediction, which involves using algorithms to predict secondary structure of a protein based on solely its amino acid sequence. So here we have some statistical or machine learning methods available under sequence-based methods. Then we have homology-based prediction methods. So these methods involves comparing the amino acid sequence of a protein to a database of known protein structures and predicting the secondary structure based on the homologous sequences or similarities. Then we have energy-based methods. These methods predict by performing or calculating the energy of each possible secondary structure using molecular mechanics calculations. There is some hybrid methods which are combination of above methods and which are termed to be more high in accuracy of predictions. One such sequence-based method today I will be discussing is about Chow-Fasman method. So it uses amino acid sequence of a protein to determine the probability of each residue being in an alpha helix, beta sheet, or random coil. This method was developed by Peter Chow and Gerald Fasman in 1970s, and it is based on the observation that certain amino acids tend to occur more frequently in secondary structure. For example, I can tell particular amino acid N is probability of being in an alpha helix is more when compared to its probability of observing in a beta sheet. So this method assigns a score to each amino acid based on its propensity to form a particular secondary structure. These scores are then used to calculate the probability that given residue is in a particular secondary structure. So how these propensities, probabilities are assigned, we'll be seeing next. So the Chow-Fasman method assigns propensity values to each amino acid residue forming alpha helix and beta strands. These propensity values are based on the observed frequencies of each amino acid in experimentally determined structures. How a propensity P of I is calculated. The propensity of a specific amino acid residue to form an alpha helix is calculated as the percentage of that residue in alpha helix relative to its occurrence in the entire data set. That forms the numerator. Then percentage. So the percentage of residue, the percentage of all residues in alpha helix, where this value represents the proportion of all residues found in alpha helix. It is computed by dividing total number of residues found in alpha helix by the total number of residues in the entire data set. So when based on these propensity values observed in the experimental structures, Chow and Fasman has classified these residues into 12 classes based on their propensity values. These classes include helix formers, strong helix formers denoted by capital H, helix formers small h, weak helix formers I, weak helix breakers with small I, helix breakers with B, strong helix breakers with capital B, as well as similarly the annotation is done for beta classes, which is shown in the table here. So before moving how assignment is done, let's try to solve a propensity value. Given the sequences and their annotation of secondary structures, can you calculate the propensity of leucine to be in the helix? The formula has been given on the left hand side. The reference for calculation has been provided on the left side screen. Okay, here is the solution. Okay, we'll try it in Jamboard. So the first is to calculate the numerator parameter in propensity. So the first thing we need number of leucines in helix. So we have 3. So 3 divided by number of leucines in a given sequence. So we have 5. So this becomes our numerator. Then number of sequences that have been predicted as helix. Number of residues in sequence that have been predicted as helix. They are 8 and total number of sequences 30. This gives you propensity of leucine to be in alpha helix. Moving forward, how this prediction rule, specific rules are applied once propensities has been calculated to predict the likelihood of helix or the strand formation within the protein sequences. The first is we have for helix prediction rules. So it has 4 steps. First is helical cluster identification. So here we try to identify the clusters of 4 helical residues, that is either denoted as H or capital H or small h out of 6 residues along the peptide chain. Wherein in case if we encounter weak helical residues that are denoted by capital I, they are counted as half. That is 2 weak helical residues are counted as 1 helix. Then we have helical segment extension. So they extend the helical segment in both direction until terminated by the tetrapeptides with average helical parameter value of less than 1. Here we have some exclusion cases such as proline, which is termed as helix breaker. Proline cannot occur in the inner helix or in the C terminal of the helix end, and we also have terminal preferences in helixes where proline, aspartic acid, glutamic acid prefer the N terminal end, whereas histidine, lysine, and arginine prefer the C terminal end. So assigning the propensities values and applying the prediction rules, a helix segment is being predicted. Similarly, strand prediction rules include strand cluster identification, strands segment extension, rare occurrence of specific residues, and terminal and charge residue preferences. Then finally we have conflict resolution. In regions with overlapping helical and strand assignments, the assignment is based on the average propensity of alpha helix or beta strand. The structure with the higher average propensity is chosen. So these rules provide a systematic approach to predict the alpha helices or beta strands in proteins, considering various factors such as residue composition, cluster formation, terminal preferences. We have other methods such as information theory and GOR. So both these methods can be used for predicting again. Information theory approaches the use of principle of maximizing the information content for a given protein sequences. The information method rely on statistical analysis of amino acid sequences and try to identify the patterns that are characteristic of secondary structures. So examples include SciSpread and JPred. Next is the GOR method, which is a rule-based method that uses a set of empirically derived rules to predict secondary structures. The method is based on the observation that certain amino acid residues have strong tendency to form certain types of secondary structures. The GOR method assigns a probability score to each possible secondary structure for each residue based on the surrounding amino acid residues. Both information theory based method and the GOR method have been shown to effective in predicting a protein secondary structures, and the choice of which method to use may depend varying on specific application. Finally, we have hydrophobicity profiles. So hydrophobicity profile is a graphical representation of local hydrophobicity along the amino acid sequences of a protein. It illustrates how hydrophobicity varies as a function of the position in the sequence. These profiles are essential for predicting various structural elements like alpha helices and beta strands. So to construct these, we need a hydrophobicity scale. The hydrophobicity scales assign numerical values to amino acids based on their hydrophobicity, and different scales exist and researchers can choose one based on their specific needs. For example, the surrounding hydrophobicity plots have been constructed by the scale developed by Ponnuswamy et al. In 1980. So when we construct a hydrophobicity profile, we plot between the hydrophobicity values of all the residues in the sequence, and we have a line parallel to the x-axis, which is an average hydrophobicity value line. When we see a pattern of alternative residues falling across this average assignment line, that is termed to as sheet, that is shown in figure 1. So if you have alternating residues along the average assignment line, that is representative for a sheet. When you have alternative residues with a periodicity of 4, here you see 2 residues are below the average line and 2 residues are above. Similarly, 2 are below, 2 are above. So this forms a periodicity of 4. So this kind of pattern represents the helix. So these methods have advantages which include simplicity and accurate prediction of turns and loops, and their ability to distinguish between the exposed and buried structural elements as well based on the hydrophobicity value assignments. There are several machine learning based methods which are recently available for topology predictions. So here I have one question. What is the purpose of Chow-Fasman method in protein structure prediction? So today the class will be for short time. The demo part of this session as well as the next session, I'll be covering it in the next session.
Sir,
Yes, yes, please.
in the previous slide,
Yeah,
this helix,
yeah.
almost all the helix in protein, always follows this pattern of hydrophobicity or not.
See, these are prediction methods. So these hydrophobicity profile methods have an accuracy of 0.69 when you use a scale that I mentioned, Poniswamy et al. That means 60% accuracy. These are just prediction methods. Prediction methods are never 100%.
No, I was under the impression only uh, helices which pack in amphiphilic or anything, it will have this kind of a feature. Whereas regular helix, which is occurring in globular protein, may not follow any of this hydrophobic profile. I
Why?
was,
Why? See, when we take a helix,
if you
we,
take a helix, there is no rule saying that one side of the helix should be hydrophobic, the other side should be hydrophilic. It should not be. There is no such rule. Uh, if you carefully observe all the proteins, it won't follow that rule. Only if it is amphiphilic or membrane bound and then there is a channel or anything, then this hydrophobic profile is acceptable for helix. This, whereas this is the general observation, but I may be wrong. Maybe you people have done some good research on this.
actually, this hydrophobicity profile has been developed by us using the scale of our prof. So here it doesn't mean falling below the average assignment line. It is hydrophilic. That is, this is not a trend like too hydrophilic, too hydrophobic. Based on the different scales, assignment scale, these assignment scales have been made such a way that positional observance along with their hydrophobic nature. So these scales come up with a positional reference as well.
Okay, sir. Thank you.
So here, as we discussed, Chow-Fasman method will try to assign secondary structure based on the propensities. So I have added some more slides which contains the quiz questions for assignment, which you guys can take it as your calculation because I have some other commitment today. So today's session will be for 1 hour. So any other questions you can ask. That's all from my side today.